Okay, let's finish up. Chapter 12, nerve tissue. This will be 203 nerve 4. And it's talking about picking up where we left off on neuromodulation. Alters the response to neurotransmitters. And can be retrograde. That is moving from the postsynaptic cell back to the presynaptic cell and facilitating synapses and strengthening synapses or inhibition of synapses, either inhibit synapses or strengthen them. And nitric oxide is one of the ones that can do this. It also relaxes or contracts smooth muscle, and then the endocannabinoids. These are, as I mentioned, we have natural opioid receptors. Now you have natural cannabinoids from the active ingredient in marijuana, and they can facilitate synapses, and they also inhibit nausea. They also make you hungry. The munchies are actually real. Endocannabinoids do make you hungry. Okay, here are those circuits, these neuronal circuits that we're talking about. So on the left with a converging circuit, and that's a bunch of inputs coming in and forming one output. And I think the book uses salivation as an example, that you think about food, you smell food, you see food, they all come together and they all converge onto salivary glands and makes you salivate. Diverging is one little input can go out to lots of outputs. I think the book uses walking as an example. So I want to walk. I'm going to fire a whole bunch of motor neurons to a whole bunch of different muscles from that one little thought, that one little input. Reverberating, thought to be either something like breathing when you're asleep, like you don't even have to think about breathing. You become unconscious and you're still breathing because we have this reverberating circuit in your brain that fires those inspiratory neurons. Or could be a memory. Memory is, so you put this reverberating circuit and its memory is in there, and so you keep on rotating your whole life until you tap into it and remember it. Then it's one strange one called parallel after discharge circuit. As you can see, there's one input, one output, but the inputs go through different routes. One of them is a direct route to the postsynaptic cell. One has two cells in between the first cell and the blue cell, and one has three cells. So what this is the effect of this is going to be one longer lasting output. So remember the speed of transmission, the slowest part of the speed of transmission is synaptic function. The more synapses there are in the pathway, the slower things go. So if you have a monosynaptic, like the first one down the middle here, is one synapse, that's really fast, causes an immediate response in the output cell. And then, but then when that output cell when the response is dying off, the second one hits because it got two cells, and so it's slower, so it gets there more slowly and continuously stimulates that output cell. And then when it starts to fade away, the one with three cells, with three synapses, then it hits. So you got this long-lasting response from one single input. And it's thought, the book says it's thought to be some of the higher brain functions like mathematical reasoning. It uses these parallel after discharge circuits. Of course, that's real speculation. Okay, memory and synaptic plasticity. Synaptic plasticity is synapses change. They're modifiable. Matter of fact, that's one of the major reasons that most synapses in your nervous system are chemical synapses because they're so modifiable. You can inhibit them, strengthen them, turn them on, turn them off. Whereas the electrical synapses, they are not modifiable. They're good for immediate response. They're really quick. And for coordinated waves of response, like in your heart. But so types of memory, immediate memory. You got to know something for, a couple seconds, and that's immediate memory, and then it's gone. Then short-term memory, it's thought to be a mechanism called post-tetanic potentiation. Potentiation is strengthening of synapses. And tetanic is really rapid stimulation. If you stimulate this presynaptic neuron, it stimulates this postsynaptic neuron. It has a certain strength of response in a postsynaptic. If you hit it several times in a row, you have an effect in the postsynaptic cell. Well, let me explain this better. So you have one synapse going off, and it affects the postsynaptic cell. Then you stop. You come back later on and do the same thing, you get exactly the same response, strength of response in the postsynaptic cell. But if you really stimulate the presynaptic cell really rapidly, really high frequency, then stop, wait a little bit, come back and stimulate the presynaptic cell once, you get a bigger response in the postsynaptic cell than you had when you stimulate it once before you did the post-tetanic, the tetany. So stimulating something, a presynaptic cell really rapidly will lead to a facilitation of synapses and to a greater response in the postsynaptic cell for a short time. So that's short-term memory. Like you got to remember something for a few minutes or so. Then long-term memory is thought to be something called long-term potentiation. You want to remember this for a long time, maybe your whole life. And there's two types, declarative, procedural. Declarative is learning facts and figures and stuff. Procedural is mechanical stuff, really, like riding a bicycle, playing the violin. You learn how to do this, and it stays with you. And this is a form of what's called long-term potentiation, that you strengthen synapses for a very long time. And this mechanism is thought to involve a couple of different kind of receptors called AMP receptors and NMDA receptors. So here, you can, even with that post-tetanic potentiation, you get a stronger response for a few minutes, but then later on it goes away. And you stimulate the presynaptic cell once, you get the same response as before the rapid response. Here, you can stimulate it for a long period, hours, days, weeks afterwards. You stimulate, you get a stronger response than you did before. So that's and what you need, we should do is go to 2-Minute Neuroscience, YouTube, 2-Minute Neuroscience. In fact, I should mention this for all of these neuro topics. It's go to YouTube and just type in 2-Minute Neuroscience, and you get all these topics. If you're unsure about one particular topic, you can go there, and one of them is on long-term potentiation. And I'd like you to, I have a link directly in D2L for the 2-Minute Neuroscience. And I think we'll pull it up right now and watch it. It's only 2 minutes. Let me pause the recording and I'll come back. Okay, I'm back. So I'm going to show it to you right here. It's like it says, 2 minutes. I'm going to pull up long-term potentiation. Oh, I got it. Yeah, I am screen sharing. Okay, right here.
Welcome to 2-Minute Neuroscience, where I explain neuroscience topics in 2 minutes or less. In this installment, I'll discuss long-term potentiation, or LTP. LTP is a process by which synaptic connections between neurons become stronger with frequent activation. LTP is thought to be a way in which the brain changes in response to experience, and thus may be a mechanism underlying learning and memory. There are a number of ways in which LTP can occur. The best known mechanism involves a glutamate receptor known as the NMDA receptor. In NMDA receptor dependent LTP, glutamate release first activates a subtype of glutamate receptor known as the AMPA receptor. NMDA receptors are found nearby these AMPA receptors, but are not activated by low levels of glutamate release because the ion channel of an NMDA receptor is blocked by a magnesium ion. If frequent action potentials cause greater stimulation of AMPA receptors, however, this will cause the postsynaptic neuron to depolarize, which eventually causes the voltage-dependent magnesium blockage of the NMDA receptor to be removed, allowing calcium ions to flow in through the NMDA receptor. This influx of calcium initiates cellular mechanisms and cause more AMPA receptors to be inserted into the neuron's membrane. The new AMPA receptors are also more responsive to glutamate and allow more positively charged ions to enter the cell when activated. Now the postsynaptic cell is more sensitive to glutamate because it has more receptors to respond to it. Additionally, there are thought to be signals that travel back across the synapse to stimulate greater levels of glutamate release. All this makes the synapse stronger and more likely to be activated in the future. This process is also associated with changes in gene transcription in the neuron, which can lead to the reduction of new receptors or modifications to the structure of the cell. These changes seem to be important for making the increased responsiveness of LTP long-lasting.
I have to stop and tell you.
So this is going to be a process of learning. Learning and long-term memory is going to be, these long-term strengthening of synapses with these AMPA receptors and the NMDA receptors. Okay, let me stop sharing that and share the PowerPoint again with you, and we'll finish up. In fact, I think we're pretty much finished here. Yeah, that's it. So that's it for chapter 12, nerve tissue. And so this is a short one, 203 nerve 4. So we'll see you later.